Greetings. My name is Amit Dhingra. I am the professor and head of Department of Horticultural Sciences at Texas A&M, and I'm very grateful for this opportunity to share some of our work. First of all, I'd like to apologize for the previous recording that had bad quality. So here it goes again. I'm very thankful to the organizers for giving me a chance to share our work. What I'm going to share today is about 15 years of work that we've developed in the area of taking sustainable approaches to producing horticultural crops in abundance. As I mentioned to you, I am affiliated with Texas A&M University. I'm also an adjunct faculty at Washington State University, where most of this work was done. I've recently moved to Texas A&M. I also have commercial interests. I have started Mu5 from my research program. I also support a couple of other companies as listed here, but none of the work or research presented here has been supported by any of these other organizations. So why do we need sustainable production today? And I think nobody is unaware anymore of the challenges that we face in production of food. All the way from weather extremes that we are facing, our soils are getting degraded, clean water is limited, as well as we need to power the planet as we are increasing our population. So how do we really make this happen has been a focus of my program. I've been working with my team over the years to really focus on the various aspects of sustainable production. We perform a lot of fundamental as well as applied research, and it's impact driven, it's need driven at times. These are some of the major areas of work in my lab. We focus on plastic biology, post-harvest quality, product health, as well as looking at plant-soil interactions. So this represents the entire value chain from the farm to the table, if you will. Of course, I want to acknowledge my team, which has really contributed to several of these projects, and I will acknowledge them, but I wanted to upfront mention that all of this work has been made possible by the support and the work of these excellent folks I've had the chance to work with. So this presentation I have 4 areas, which goes all the way from soil to post-harvest. So it represents this continuum of the value chain. I'll touch upon these topics. Of course, these are in a short time, you can't go in much detail, but I'd be interested in collaborating and interacting further with colleagues who might be interested in these different areas. So I'll start with the first part of the soil microbiome interaction. In 2015, I got involved in some research with biochar. Biochar is produced as a product of pyrolysis, where you burn biomass at high temperatures and low oxygen, and it creates this carbon soot-like material which you can then incorporate in soil. So we've done many trials, and this example exemplifies what we have observed over and over again. When we add biochar into the soil, we see this massive increase in vegetative growth. These plants were planted at the same time. And we've seen a lot of increases in yields. We've done trials across different crops. Of course, permanent crops are missing in this right now, but we are doing those trials as well. However, you can see some massive increases over controls. And the question then becomes, how is biochar working in the soil? And there are several hypotheses around that. Some of them have been tested and shown to be working. So the biggest part is that biochar retains water in the soil, and it also its absorption power allows for holding onto the fertilizer, the water. Also, it improves soil structure. It changes the bulk density. It enhances microbial activity. So we were really interested in seeing how this is happening, and we've done a time course analysis of the soil rhizosphere. But the biggest challenge before we apply the biochar is, where do you get consistent quality biochar? And what you're seeing here is actually my spinout, which has one of the sections is to produce biochar, and the biomass processing units are able to produce a consistent quality biochar over and over again. So we did this project with the biochar obtained from Mu5, where We looked at the root system as well as the associated rhizosphere or microbiome associated with the root system. This project was funded by SARE and at WSU. And what we found was very interesting. That at four different developmental stages, in the control samples, the microbes keep on increasing over a period of time. Now recall that microbes are a sink for carbon. Whatever carbon that the plant is fixing is also being consumed by the microbes. Very interestingly, in the presence of biochar, the maximal growth of microbes stops at the fruiting stage and it drops during mature stage. So this is also an indication that we are seeing that there may be a better communication with the microbes and the plants that is happening thanks to the biochar. So it's fascinating to see. We're working on wrapping up this work right now. I do want to say that these data were generated using RNA from soil, not DNA, which is commonly used. Our goal is not only to find who is there on the microbe identity part, but also which microbes are functional. So this is a pretty cool insight that we are starting to gain with this work. Next, I will jump into the topic of plant-microbe interactions. So it's great that the plant and microbes can interact, but also product quality is very important in the sense that we need to be able to grow plants that are healthy. And soil-borne diseases are a big issue. And one of the issues that our lab has worked on and trying to understand how the interaction happens is root rot in peas. We've done a pretty cool approach of combining genetics and gene expression-based approaches to identify some novel QTLs, quantitative trait loci, where we've identified additional genomic regions that are probably interacting with the Fusarium solani in the soil, the peas, which gives us additional insights into how the interaction may be happening. In addition, we have found several genes that respond to this challenge by the Fusarium root rot pathogen. And in those genes, we found some alleles, some targets that we can then either edit or create transgenic pea to be completely resistant to root rot. For that, you need a very robust plant transformation system or pea transformation system, which was developed by our graduate student, Bruce. And he's been able to demonstrate that he can generate, he can really do a lot of transgenic peas, highly efficient method, and we've already utilized it to create gene editing as well as transgenics. So we took one of the anthocyanin biosynthesis genes as a target where we overexpressed the gene and silenced it. Because of perhaps epistatic interactions, we don't see much phenotypic differences. However, very interestingly with CRISPR, we are able to obtain colorless flowers. We've been able to knock out the anthocyanin biosynthesis gene from these bees. And here's the proof where we have identified lines where the genomic area for this particular gene has been edited. So with this type of a platform, now we can utilize this for gene editing, for expressing other type of proteins in peas. And pea is very important, of course, not only from the fact that Mendel used it, but for genetic studies, but also it is the new source for alternative protein. So again, going after that sustainable, nutritious food concept. Now I will switch to pre-harvest quality, which is in this particular area, we are really looking at something novel, which is when you look at all the fruits and vegetables around you, flowers even, they represent this myriad colors. Now, these colors are not just anthocyanin, which is a vacuolar pigment. Several of these are xanthophylls, carotenoids, and various other pigments, which are localized in the plastid or the chromoplast. Now, everybody's familiar with green plastid, which is the chloroplast, where photosynthesis happens. But now we are really interested in the biology of the chromoplast across diverse fruit systems. The challenge is we just can't go in and look at the chromoplast because it's hard to isolate these chromoplasts and do proteomic studies. Most of our studies have been focused on tomato, where chromoplasts have been characterized, but there is such a diversity of fruits and vegetables out there. Why is that important? It's not only for the pre-harvest, how are these fruits growing in the field? How do they deal with extremes of temperature? How do they deal with extremes of light conditions and other environmental conditions? also biotic issues. We can learn so much more from these plastids. So we did a microscopy characterization of apples, three types of apples, which are red, top red delicious, anthocyanin predominant, pigment predominant apple. Granny Smith has more chlorophyll. And then finally, golden delicious, that is xanthophyll predominant cultivar. What we saw that the diversity of plastids was amazing. There were multimorphic plastids present depending on the type of apple and at different developmental stages, as well as just within the epidermal and parenchymal cells. We just saw amazing diversity of plastids. And then as the fruits mature, you start having, depending on the variety and depending on the cell layer, you have different types of metabolic structures appearing. Now, this is great. We have visual, and we want to know what is the content of these proteins in the plastids. For the uninitiated, while there are about 30,000 genes in the nuclear genome, there are about 130 proteins already being produced in the chloroplast. But the chloroplast imports anywhere from 3,000 to 5,000 proteins from the cytosol to carry out all the metabolic functions. And through our bioinformatics predictions, we have identified the best combination of predictive software to identify species-specific plastid-targeted proteins. And this has given us a fundamental sort of a database which we can compare to. These are all predictions. Those are great. We need tools to now go and do proteomic analysis. And recently, one of my grad students in the program, June, has developed a method of isolating plastids from fruit, different type of fruit. And what we observe here are these multimorphic plastids. These multiple bands represent the multimorphic plastids we saw in the microscope. So we've made progress in the technology to really get to a point where now we can perform proteomic characterization, identify the genes that may be responsible for aroma, for health, for disease. So that this is really exciting phase of the project. And one of the applications of this is that when we protect the degradation of these photosynthetic membranes, we can keep the fruit healthier on the tree longer. That means the fruit increases in size, it fetches more money for the farmer, as well as during storage, fruit really stays of higher quality during storage as well. Finally, I'll switch my topic to post-harvest quality, where I want to acknowledge the work of four of my lab members here. And what we started working was with pears. Pears, this is a typical form of ripening that we characterize in fruits, climacteric versus non-climacteric. I do want to comment that it's a spectrum. It's not these two boxes, but in climacteric fruit in general, there is a concomitant increase in ethylene as well as respiration rates, whereas that doesn't happen in non-climacteric fruit. But in pears, which are a member of the family where apples, the rose family belongs, apples, roses, almonds, peaches, nectarines, they all belong to the same family, cherries. Pears are also climacteric. However, they're picked mature but unripe. Apples are picked mature and ripe, and the best way to test is eat the fruit off the tree. Apples will taste sweet, pears will taste starchy. Interestingly, European pears, not Asian pears, but European pears require a certain amount of cold exposure post-harvest before they can ripen. So that's very fascinating. And this duration of cold exposure is quantitative. It's genetically regulated. One cultivar, such as Bartlett, would require 15 days of cold exposure, whereas an Agile pear can require up to 60 days. And one other conundrum is that if you apply this ethylene receptor blocker, 1-methylcyclopropene, the pears don't ripen, but apples ripen even after application of 1-MCP. So it's really an enigma of why this happens. So just to revisit, in pears, the ripening is a little bit unique. It requires this pre-ripening period of cold conditioning, which is unique. And we were interested in understanding what is happening during this cold conditioning to all ripening-related genes. And based on our first study, we found out that, very interestingly, the operative oxidase gene had heightened expression at 100% conditioning, which is when it has obtained all the code it requires. And the cool part is that this was this AOX overexpression was pre-climacteric. AOX is involved in ripening, and it's usually seen at climacteric or reach the highest level of expression at climacteric or post-climacteric. So AOX really has a lot of roles that it plays in the mitochondrial cytochrome C pathway. It's an alternative to the cytochrome C pathway. And it also perhaps transmits retrograde messages to the nucleus to signal stress. So cold does induce AOX, we know that in general, but in this particular post-harvest condition, that was a very unique observation. And this is where we've done a very detailed analysis of two different cultivars, Orchid and Anjo, exposed them to cold, and did the RNA-seq work, and found out that there were different alleles that are involved in these two, and they might be the ones responsible for the need for different amounts of cold to induce the ripening pathways. We used this AOX method to see if we could activate AOX in the 1-MCP treated fruit and ripen the fruit as well. And interestingly, we were able to do that. Right now, Evan Stowe, who's a graduate student in the lab, is trying to understand if we apply the AOX activator glyoxylic acid from the outside of the fruit, how does it activate ripening? GLA is supposed to activate the glyoxylate cycle or the mitochondrial TCA cycle, and how does it result in the increase of ethylene? Some of these questions is what we're studying. Now, we've taken this concept and already applied to produce fresh sliced pears. This is a new product that can really provide a lot of economic benefits to the farmers. So in the end, I would like to acknowledge the funding from various groups that has supported the work that I just showed you. If there's any interest in any of these areas, I would love to connect with you. Thank you so much again to the organizers for this opportunity to share our work, and look forward to further interactions. Thank you.